good afternoon and welcome to the College of the Environment Environmental Speaker Series. As usual, please put away those laptops, tablets, cell phones, turn them off, put them away so you can just enjoy the presentation with your full undivided. Those of you at home on Zoom, no, you can't turn away your computers. You have to keep on your, that's different. But in the room, we'll keep it screen free if we can. As usual, we will have time for questions at the end, both in the room and over the Zoom. Those of you in the Zoom can submit questions via the chat, and we'll get to as many as we can. My name is Stephen Freeland. If you have any questions, you're always welcome to email me as well. So our guest today is Brooke Weigel, and Brooke is out at the Friday Harbor Labs, part of UW, out on San Juan Island, just near Friday Harbor. But she's been doing a bunch of other cool stuff before she got there, studying sponges down in North Carolina. And the coolest PhD field work that I've come across yet to date. She spent five summers out on Tatoosh Island. You know where Tatoosh Island is? Few of you, it's the coolest. Once they open up again, and you get to go out to Nia Bay, Cape Flattery, which is one of the high points on the planet, and you look out. This is the north and westmost tip point of the continent, of our part of the continent. You look out over the water, and there's this little island out there with just these crazy wet waves, and there's a little lighthouse that used to be there. That's Tatoosh Island, part of the, right off the Macaw. I don't know if it's actually
part of the Macaw tribe. Yep.
Super cool place to do your field work, which we'll hear about in the context of kelp and other good stuff. Thank you, Brooke. Welcome.
Yeah, thank you so much for the introduction. Can everybody hear me okay in the room and over Zoom? And welcome to the Zoom audience as well. It's really nice to be here. I appreciate you guys all coming on such a sunny afternoon after a whole week of rain. And it's also just nice to be here talking to you guys in person because I've been mostly giving Zoom seminars for the last 2 years. And so today I'm going to talk to you a little bit about my postdoctoral research studying the effects of ocean warming across the life cycle of bull kelp.
I'm getting that.
Are we
Yeah.
almost ready? Okay.
I think you should have control now. Yes.
Awesome. Okay. So for those of you who aren't familiar with kelp, kelp are brown algae in the order Laminariales. They are evolutionarily distinct from red algae and green algae in plants. And there are 26 species of kelp in Washington state, but we have only 2 species of what we call canopy forming kelps, which are the really massive kelps that grow from the bottom of the ocean all the way to the surface. So in Washington, we have Nereocystis pyrifera, the giant kelp, which also grows down through California. But in Washington, it's restricted to the outer coast. So you'll see it out on the Olympic Peninsula, and it goes into the Strait of Juan de Fuca a little bit. And then we have the bull kelp, Nereocystis luetkeana, which is the dominant canopy forming kelp that grows throughout Puget Sound and the Salish Sea, all the way from Central California through the Aleutian Islands in Alaska. So kelp are foundation species and ecosystem engineers. They are the trees of the underwater forest, and they provide not only structural habitat, but also food and resources and shelter for so many really economically and culturally and ecologically important species like salmon and rockfish and marine invertebrates. And people sometimes ask me, what's the big deal with kelp? What happens if we lose the kelp forest? And I tell them to think about if you have a forest, but you lose all the trees, it's a big deal. So kelp are really important to these underwater forest ecosystems. And I've been studying kelp for about 7 years. My dissertation research was focused on the kelp microbiome and carbon cycling in kelp forests. So I was studying the millions of bacteria that cover the slimy photosynthetic surfaces of kelp. And this is an actual microscopy image of what those bacterial communities on the kelp look like. They're not actually colored. We colored them so that you can see the cells. And I was also studying carbon fixation because kelp are among the fastest growing primary producers on the planet. They can grow multiple centimeters per day, and they fix more than 2.35 kilograms of carbon per meter square of kelp forest per year. And I found out that they're also leaky. They leak some of that carbon into the seawater as dissolved sugars, which has important interactions with their bacterial community. And I'm not going to focus on this research today, but if you're interested in microbes, we can talk about that later. So where are kelp? They are distributed globally in temperate and Arctic oceans. We have a really high diversity of kelp here in the Pacific Northwest, and kelp are also present in the North Atlantic, in the South Atlantic, over in Japan, they're in South America, South Africa, Australia, and New Zealand. So kelp are pretty much everywhere in very cold waters. And unfortunately, kelp are in decline in many locations worldwide. And this is just a map showing different potential stressors contributing to kelp forest declines, such as warming, heat waves, pollution, eutrophication, and a variety of other factors. And kelp have been very popular in the media lately because of these declines. There's a lot of articles that are being published about kelp. You might have read some of them online yourself. There has been a particularly bad collapse of kelp forests in Northern California that's pretty linked to the loss of sea stars from sea star wasting disease. And sea stars are voracious predators of sea urchins. So when you lose the sea stars, the urchin populations increase and the urchins decimate the kelp forests. So again, there's kelp declining in different places around the world, but likely they're declining for different reasons in different places. So what are some of the factors contributing to declines in kelp forests? Well, some of the known contributors are thermal stress from rising seawater temperatures, overgrazing from predators like kelp crabs and sea urchins, the loss of keystone species like the Pycnopodia sunstar, which drives up the population of urchins that eat the kelp, things like pollution and sedimentation, and perhaps other environmental stressors. My postdoctoral research is funded directly by the state of Washington to really understand in our local waters what is happening and why are these kelp forests declining. So I'm going to be focusing my talk on the bull kelp, Nereocystis luetkeana, as it's the dominant canopy-forming kelp species in Puget Sound and the Salish Sea. This is what they look like from the surface of the water. I'm guessing many of you have seen that if you've gone out on the water in the state. And they look really incredibly beautiful from underwater as well. So as I've mentioned, bull kelp forests are declining in some areas in Washington state, particularly in South Puget Sound. So here's a map of, here's a map of the Salish Sea. So we're up here. This is Puget Sound. And this area, region of Puget Sound called South Puget Sound is really adjacent to Olympia. And my collaborator, Helen Berry at the Washington DNR, along with others, have found that over the last century, we've lost 60 to 80% of kelp in southern Puget Sound. So this map shows the presence of historical kelp along shorelines in blue. And then these two pink areas, the one I've circled here and over by Tacoma here, are the only two places where kelp still exist in southern Puget Sound. So we have lost a lot of these kelp forests. And places where we've lost kelp tend to have really high summertime water temperatures that can exceed 12 to 15 degrees. And they also have really low dissolved inorganic nitrogen concentrations. Now, this is kind of counterintuitive because you might think that closer to urbanized areas, there would be a lot more nitrogen. But what happens during the summer is the water gets so warm that it stratifies and forms a warm stratified layer at the surface. And then in that stratified layer, all of the nitrogen gets depleted by plankton and algae. And so you end up with this warm, nutrient poor layer of water that's not particularly great for bull kelp since they require a lot of nitrogen to sustain their fast growth rates. And this is just a model from Parker McCready's group at UW in the coastal ocean modeling group showing sea surface temperatures for August 2011. So not 2011, 2021. Here we are. And you can see that the water temperatures in southern Puget Sound and really throughout the Puget Sound Basin get quite hot, up to 18 or 20 degrees Celsius. And in contrast, I'm working up on San Juan Island, which is right here up at this blue area at the top of the map. And the San Juan Islands are generally much cooler, although you do see some warm water influence from the north coming from the Fraser River outflow. And even though temperatures are rising throughout Puget Sound, and when I started this postdoc, I thought, oh, the San Juan Islands, those are so cold. It's always cold there. I've gone diving, it's very cold. But it turns out that the subsurface temperatures are rising there too. Graduate student Kendall Muri has been doing a lot of diving to monitor the temperatures in the San Juan Islands. And this is some of her data from last summer showing that in the kelp forest, temperatures within the kelp forest exceeded 16 degrees on more than 10 days in August. And they even exceeded 20 degrees. So a lot of the surface waters are warming, and we need to understand the implications for bull kelp. So my main research question is to understand how warming ocean temperatures impact bull kelp across different life history stages. I'll tell you about the kelp life cycle in a minute, but there's a few different stages of bull kelp. You are used to seeing the large kelp that reach the surface of the ocean, but there's also little juvenile kelp that are actually starting to grow right around now in the springtime. And then kelp have a weird life cycle with these tiny microscopic things called gametophytes that also overwinter on the bottom of the ocean. And water is generally colder at the bottom than it is at the surface. So those different life history stages might be experiencing different temperatures. This is the life cycle of the bull kelp. It's pretty cool. There's the adult sporophyte here, which is the giant kelp that you see out in the ocean. And towards the middle and end of the summer, they get these dark patches on their blades called sori. And these are the reproductive patches, which contain millions and millions of spores called zoospores, which these patches kind of like fall out of the blade like cookie cutters, or the whole blade falls off. And then they release these zoospores, which are microscopic, but I'm drawing them here so you can see them. And the zoospores settle on the bottom of the ocean and germinate into separate male gametophytes and female gametophytes. And these are also microscopic. And these gametophytes live on the bottom of the ocean from like the midsummer all the way until the next winter and spring when they germinate. So they are the stage of this kelp that overwinters. And this kelp has what's considered an annual life cycle. So just like an annual plant, the adult sprouts out once a year. And we start seeing these juveniles, which are formed when the male gametophyte fertilizes the female, and it grows into a small baby kelp, which gets bigger and bigger until it forms these giant kelp, and they reach 100 feet tall by midsummer. So it's really an incredible life cycle. It's also like if your eggs and your sperm lived outside of your body for like a year without you. So it's pretty weird that they have this amazing life cycle, and I'm excited to be able to study it. So I'm going to focus the first part of the talk on the effects of temperature on the gametophytes, the microscopic form of the kelp. And so my research question is, how does temperature impact the growth and reproductive success of bull kelp gametophytes? What are the thermal tolerance limits or the maximum temperature that they can withstand before dying? And are there optimal temperatures for growth and reproduction of the gametophytes? So I go out on a boat, I collect reproductive sori patches from the bull kelp, I bring them back to the lab and release the zoospores. And this is what the zoospores look like in solution. They're microscopic, so it just looks cloudy, but they release millions and millions of these little zoospores. I put the zoospores into 7 different temperatures, and these temperatures are 10, 12, 14, 16, 18, 20, and 22 degrees Celsius. So I'm covering sort of the whole range of temperatures that they might be experiencing. 20 and 22 degrees are pretty extreme, especially for bottom temperature. But as we see in a warming world, it's good to sort of look at the more extreme temperatures and see what happens. And so I have them growing in these little tissue culture plates in aquaria that I set 2 different temperatures in a cold room. And in addition to just looking at what temperatures they can survive at, I'm interested in looking at variation across populations, because I showed you that the temperatures can vary widely across Puget Sound. So I'm growing kelp from multiple different populations to try to understand if there's populations that are more adapted to these warmer temperatures and can survive better than other populations. And it's important to know this because groups like the Puget Sound Restoration Fund are actively growing and outplanting kelp to do restoration. And so we need to know like which population to select in order to grow these kelp. So for my first summer of experiments, I was growing kelp from Tern Rock in the San Juan Islands, which is a relatively cold water population, as well as Squaxin Island in southern Puget Sound, which is one of those populations that's in decline and it experiences much warmer water. And then next summer, I hope to grow kelp from additional populations in between. And I would like to acknowledge the Squaxin Island tribe for giving me permission to collect kelp on their land, as well as the Washington DNR for facilitating boat access to kelp. So after I grew these kelp in the lab, the way that I measure their growth and survival is simply by putting them under the microscope and taking photos every 5 to 10 days. And I take 3 random non-overlapping fields of view per well. And then I can look at the photos and assess how the gametophytes are doing over time. So pictures speak louder than words sometimes. And this is just a photo from one of my first experiments where I could really clearly see the effect of temperature. And what's interesting is that at 10 degrees, the coldest temperature, growth was actually slower. So metabolic rates are slower at colder temperatures, but then they grew the best sort of in these mid-range temperatures. But by 20 and 22 degrees, you start to see deleterious effects, which I'll show you more graphs in a minute. And my first experiment was rather dense, so it was difficult to measure individual cell sizes. So I quantified them by just taking the total % area of the field of view of the microscope as a proxy for biomass of gametophytes. So I'll show you the first result comparing those 2 populations. The warm water population from Squaxin Island is in orange. The cold water population from Tern Rock is in blue. And the first thing to note is that the gametophytes actually were able to grow across a range of temperatures, including up to 20 degrees, which was surprising. But what's also really interesting is, so we have temperature on the x-axis and % area of gametophytes on the y-axis. And you can see that the warm water population from Squaxin Island had a higher density of gametophytes at some of the warmer temperatures than the cold water population. And if I graph it a different way, looking at across time on the x-axis and the percent area of gametophytes on the y-axis, you can see that across all the days, the warm water population had the highest amount of gametophytes at 16 degrees, while the cold water population had the most gametophytes at 12 degrees. So there do seem to be some population level differences in their ability to grow at these warm temperatures. And just to show you some of those results in pictures rather than graphs, you can see the difference between the populations actually visually. So this is photos across temperatures. I don't know if you can see, but they're 10, 12, 14, 16, 18, 20, and 22 degrees. And you can see that at 20 and 22 degrees, the cells actually become colorless. They lose their pigment and they pretty much die, which is a bad thing. And these are from the cold water population in the San Juan Islands. And if you compare that to gametophytes from the warm water population, you can see that at 20 degrees, these guys still have their pigmentation. So that's really interesting because it does look like the warm water population might be able to withstand slightly warmer temperatures than the cold water population. So again, that's cold population, warm population. But neither of them are able to survive at 22 degrees. So that was like the first experiment I did, and I wanted to do another experiment at lower density to see what the effect is on fertilization success and production of those baby sporophytes. So I repeated this experiment with the cold water population from Turned Rock at 2 different densities. And I did the same gradient of temperatures from 10 to 22 degrees. And these were, I should mention that this experiment, they were germinated, the spores were put directly into those experimental temperatures, whereas the last experiment, I allowed them to grow for 10 days at 10 degrees Celsius, giving them like a head start. But now I wanted to see if you release the zoospores directly into the warm water, do they survive? So this is a graph with temperature on the x-axis and the percent area of gametophytes on the y-axis with 2 different densities. And you can see the first thing to note is that none of them germinated at 22 degrees. So 22 degrees seemed to be an upper limit for the germination of zoospores into gametophytes. And what's also striking is that they grew with a pretty high density at 18 degrees, but then between 18 and 20, they really like tanked. So it seems like gametophytes are actually fairly hardy in that they can germinate and grow at some pretty warm temperatures, but it's bad news once you hit 20 degrees. So next I'll show you results from that experiment of the baby sporophytes. So this is what it looks like when the gametophyte male fertilizes the female and forms a little baby sporophyte. They're super cute to look at. And what I found in this experiment for sporophytes is that sporophyte production stopped above 16 degrees. So there were juvenile sporophytes at 10, 12, 14, and 16 degrees, but there were no sporophytes at 18 degrees or at 20 degrees, and the 22 degrees never even germinated. So this is really important because obviously to have a healthy kelp forest, you need to complete the life cycle. So it seems like the gametophytes can maybe withstand some slightly warmer temperatures, like up to 18 or 20 degrees, but they will not have successful fertilization and turn into baby sporophytes at temperatures higher than 16 degrees. And I do need to repeat these experiments this coming summer across more populations because that temperature threshold might vary. So to conclude the gametophyte part of the talk, they germinated at temperatures from 10 to 20 degrees, but did not germinate at 22 degrees. I saw deleterious effects at 20 and 22 degrees. The cells became colorless and they sort of died and growth slows. And I found evidence of population specific temperature tolerance. So the warm population had higher growth rates at 16 degrees, and the cold population did better at 12 degrees. But sporophyte production or sexual reproduction stopped above 16 degrees. Okay, so now I will transition to the second part of the talk, looking at how heat stress and nitrogen limitation impact bull kelp adult sporophytes. So next, we're going to focus on the big guys because these are hanging out in the water, and the adults are experiencing probably a warmer thermal regime because they have this gas-filled float that keeps them at the surface, and the blades are sitting near the surface of the water where it tends to get very warm. So we want to know how temperature affects those adults as well. And it's difficult to study this because in the environment, temperature and nitrogen are often correlated. When you have upwelling of water in the ocean, that water is typically very cold and nutrient rich. And then in places like South Puget Sound that don't have a lot of exchange with the open ocean and have shallower water, you tend to have warm, nutrient depleted water. So warm water tends to go with depleted nitrogen and vice versa. So in order to study the different effects of temperature and nitrogen, you need to do an experiment in the lab, which is what we did. So the questions for this part of the talk are, how do elevated temperatures and low nitrogen concentrations affect the physiology of bull kelp sporophyte blades? Sporophytes are just those adult kelp. And do low nitrogen concentrations exacerbate the stress of high temperatures? Are they more stressed when they lack nitrogen? And these experiments are done completely collaboratively with graduate student Robin Fales, who's a PhD candidate in Emily Carrington's lab at UW. So it was really fun to work together on this project. So we did an experiment at Friday Harbor Labs manipulating both temperature and nitrogen. We had 3 different temperatures, 12 degrees, 16 degrees, and 20 degrees. And then at each temperature, we had 2 different nitrogen concentrations. We had a low nitrogen concentration of around 2 micromolars of nitrate and a high nitrogen concentration of around 30 micromolars of nitrate. And to put that into context, southern Puget Sound gets to below 2 micromolars during the summer, and 30 micromolars is like a good upwelling day on the outer coast. So that's sort of the full range that they might experience naturally. And we had replicate kelp 6 blades in each tank in these recirculating water tanks. And we measured a variety of factors to assess temperature and nitrogen stress on their physiology. We measured the physiology of their photosynthesis using PAM fluorometry. And we did that daily for 10 days. We also measured at the end of 7 days of exposure to these treatments, we measured photosynthesis and cellular respiration rates using a sensitive oxygen electrode and blade tissue plugs, so small pieces of the blade. And then finally, we measured nutrient uptake rates of the blades after 8 days in these experimental treatments. And to begin, I'll show you a picture of the kelp blades at the end of this experiment. And you'll see that in our most stressful treatment, the 20 degrees without additional nitrogen, those kelp like physically dissolved. So they actually died, and we weren't able to assay photosynthesis, respiration, or nutrient uptake from those kelp because they were physically dissolved, which I think speaks to the stress of having high temperature and low nitrogen. I will note that these are the actual measured temperatures in the tanks. Our cold treatment was 11.8, our medium treatment was 15.9, and our warm treatment was 20.6. And it's a little higher than 20 because one of the chillers went out for part of a day. So that might be a bit higher of a temperature than we anticipated. I should also note that the kelp were healthy, at least not this treatment, but the other ones grew like 10 to 20 centimeters throughout the week. So they were doing relatively well in the tanks. And this is a graph of the photosynthetic fluorescence. It's basically just measuring the condition of the kelp blades and the condition of their photosystems. And you can see that all of them had relatively good conditions until the 20 - N treatment like tanked right before those died. And here's a graph showing the net photosynthesis rates. So this is oxygen production of these different kelp blades during photosynthesis. And on the x-axis, we have different treatments of 12 degrees minus nitrogen, 12 degrees plus nitrogen, 16 degrees minus nitrogen, 16 degrees plus nitrogen, and then the 20 degrees. And basically what we found is that at 20 degrees, there was significantly lower photosynthetic rates. We also lost that one treatment, so we only have one of the 20-degree treatments, but the short story is photosynthesis rates dropped at 20 degrees. And then when we looked at respiration rates, we saw the opposite trend, that respiration rates were higher in the 20-degree treatment, and that indicates like elevated metabolic demand, which makes sense. This trend wasn't significant because there was a lot of variation, but I think you can still sort of see that trend happening. And then finally, we measured nitrogen uptake rates by the kelp blades at the end of the experiment, and we found that the nitrogen depleted kelp had significantly higher nitrogen uptake rates, which basically just indicates that our nitrogen starvation treatment worked, because when we measured nutrient uptake rates, we gave them a pulse of nitrogen, and the ones that had been starved were really greedy and they took it up quicker. So this just shows that they were nitrogen starved during the experiment. So to conclude the adult kelp experiment, I think the most important thing is that we found no deleterious effects at 16 degrees compared to 12 degrees. So all of our physiology measurements didn't show differences at 16 degrees, which we thought that they might, because 16 degrees is a relatively high temperature. So that's actually good news for the adult kelp. But we did see deleterious effects at 20 degrees. We saw blade tissue degradation and loss in the low-end treatment, and we saw elevated metabolic demand. The 20-degree kelp had higher respiration rates, and they had lower photosynthesis rates. So they're not able to keep up with their energy demand. We saw higher nitrate uptake rates from the end-depleted kelp. And in this experiment, we didn't see any interactive effects of temperature and nitrogen statistically, but that's because our most stressful treatment died. So again, we need to repeat this experiment next summer. And we're going to be repeating it with bull kelp exposed to different temperature and nitrogen regimes from across Puget Sound. So I'm just about to start my spring and summer field season in about a month. And I'll be doing more of these gametophyte temperature tolerance experiments with bull kelp from additional populations in the Salish Sea. I'm also going to look at the effects of nitrogen as well as temperature on the bull kelp gametophytes, because the media that everybody grows kelp in is super nitrogen rich. The concentration of nitrate is 800, which is a lot. So it's pretty much every kelp growing experiment is done at nitrogen concentrations that are not at all realistic to what's happening in nature. So I want to do some nitrogen experiments to see what the effective temperature is when they have a nitrogen concentration that they might actually experience in Puget Sound. And then I'm also going to repeat the sporophyte temperature and nitrogen experiments with bull kelp from additional populations. And then hopefully Robin Fales and I will compare the effects of temperature on bull kelp versus an understory kelp called Saccharina latissima, which grows, it doesn't get as tall as bull kelp, but it's present in many locations in Puget Sound. With that, I would like to acknowledge many people who made this research a success, especially the Washington State Legislature for funding the Kelp Conservation Proviso, which funds my research. My advisor, Dr. Megan Detier, Robin Fales, the graduate student who's helping me with a lot of the sporophyte experiments, as well as many other people as part of the Friday Harbor community. I'm grateful to everybody. All right, and I think we have time for questions.
Great. Question. You do it, right?
Yeah.
So on the experiment that you said, like, it got warmer than it should have been, for what was the reason?
Yeah, so it's really hard to make seawater cold. You need a chiller, which is like a little AC unit that the water runs through. And we were just starting this experiment, and like some of the equipment was hadn't been used in a few years. So one of the chillers like broke for a little while, but we got it back online right away. So the temperature did get a little warmer than we wanted to, which is why I'm definitely going to repeat that experiment next summer.
Okay, so when it was saying like ±1.4, do you think so at some point it probably reached about 22 rather than 20?
Yep. Yeah, it was definitely warmer than we had anticipated, although 22 degrees is also a temperature that is measured at the surface in Puget Sound, so it's not like totally crazy. Yep.
I have someone from Zoom. They're also requesting if you could please repeat the questions. So that people on Zoom can hear what you asked.
Yeah.
But the question I have is regarding the warming of sea temperature in areas that have lost kelp forests. Do the kelp aid in stabilizing the sea temperature?
That's a good question. So they asked if kelp helps stabilize the sea temperatures in warming areas. I would say no. Kelp actually can locally increase the temperature because they like slow the movement of water and kind of retain water. And my PhD advisor, Kathy Pfister, has a study looking at the temperature inside and outside of kelp forests, and the temperature is always a little warmer inside the kelp forest.
You mentioned that the kelp, like, so they start out as a little tiny like microscopic guys. You said that they grow like 100 feet. Like, is that like in just like one growing season that they grow that much?
Yeah, so the question was, kelp start as little teeny microscopic things and they can reach 100 feet tall. Does all that growth happen in one season? The answer is yes. So this kelp start growing right around now. You'll start seeing little juveniles like a few feet tall, and they actually can reach 50 to 100 feet by like July. So they grow extremely fast. Yeah. Yeah, so what is the kelp's lifespan without the temperature effects? So this species is an annual kelp, so it does naturally die off within a year. Sometimes they overwinter and hang out for 2 years, but it usually just is there for a year. And the reason is that they kind of grow throughout the whole summer, and then into the fall, they release all those spores and their tissues kind of like shred. They become like really broken. And then in the fall, the waves and the storms start hitting them, and they actually just get ripped out by the big winter storms, which is why you'll see like giant piles of kelp on the beach in the fall and winter. But the giant kelp, which is the other species, that one is a perennial. So that one actually does live like 2 to 6 years. Yep.
And interesting to see that out there, go rubber, seal it down, just can't see the end of it. It was really interesting. Another question I had, do you actually stay on Catfish Island? Or you go around?
Yeah, so somebody had a question about Tattoosh Island. He's been out there fishing. And yeah, so I was working for my PhD with Kathy Pfister, who's been working out on Tatoosh Island for the past 30 years. And we work with the Makah tribe who owns the island. So with their permission, we would go out and like sort of camp on the island during the low tides every 2 weeks throughout the summer. Oh, nice. Yeah, if you go to the hike from Nia Bay, you can like look out at the island. If you see people in orange overalls, that's us. Yep.
There's a couple more questions online about the temperature. Is there any speculation on episodic stress, like from the recent heat wave? And also, is there any way of knowing how long it takes for kelp to adapt to warmer temperatures?
Yeah, so someone had a question about episodic stress, especially during the heat wave. Yes, the heat waves are definitely really bad for kelp, especially at the surface. So I actually started this research in July. So I got here like right after the heat dome. So I didn't actually witness firsthand the effect of the heat dome, but some of my lab mates in Emily Carrington's lab documented the effect of the heat dome, and it killed off a lot of intertidal algae, but some of those like Hedophyllum were actually able to recover. So they kind of died back and then recovered, but it definitely killed off a lot of intertidal species and generally is not great. We're going to keep monitoring. Obviously, if there's a heat wave next summer, I'll be there documenting it. And then what was the other part of the question?
Is there any way of knowing how long it takes for kelp to adapt to warmer temperatures? And how does that compare to the current rates of growth?
Yeah, that's a great question. So somebody asked, how long does it take the kelp to adapt to these warm temperatures? I would think a pretty long time, like on evolutionary timescales, probably much longer than the scale of warming that we're experiencing. Another thing to note is that these different kelp populations I'm sampling, somebody has shown that they have distinct, like their genomes are slightly different. So they have, genetic variation that might help them adapt to the warm water. And we don't really know enough, like the field of kelp genomics is just starting. So we don't really know that much about, what genes are necessary to adapt to warm water and things like that. But that's like a really active area of research, hopefully into the future. Yeah.
Could in the future, kelp that is more heat resilient, how successful are transplants to areas that are becoming warmer?
Yeah, so you had a question about resilient kelp being transplanted out into the water. And I will say that this research is also sort of in its infancy, but Puget Sound Restoration Fund is a nonprofit that's actively growing kelp and outplanting it at a site in Puget Sound. And they do they plant basically little baby sporophytes that are a few centimeters tall. And by the end of the summer, they get a giant tall kelp forest. But I don't think they've been able to successfully have those kelp reproduce and form their own subsequent generation. So there's still like some research that needs to be done. And I don't think we've experimented yet with planting any resistant populations because we're sort of just starting this work on figuring out how populations might differ in their response to temperature. But that's definitely the next step, would be like, using that to inform management about what populations you should select for growing. Yep.
So you're talking about how across the world there are a variety of reasons why kelp are defined like area.
Mm-hmm.
What degree of confidence, are we confident that in our area, Puget Sound, that it is in fact heat and not other environmental stresses that are the main components?
Yeah, so the question was, kelp are declining globally for a variety of different reasons. How confident are we that in Puget Sound it's related to temperature and not other stressors? I would say that we So the research that the DNR has done has sort of identified warm temperatures and low nitrogen concentrations as correlated with places where we've lost kelp. So we know that this has also been demonstrated in British Columbia by a lot of folks up there. There seems to be a trend emerging that the places that we lose kelp have warmer water and less fresh seawater, and they tend to be like shallower, more protected areas that don't get as much wave energy. And so there seems to be a lot of evidence pointing to the fact that it is likely linked to temperature and perhaps also nutrients. And so that's sort of the first place that I'm starting with this research is, temperature seems to be the obvious stressor. But there could be other stressors as well. It could be like sedimentation near urban areas, could be blocking the light from gametophytes, or like, different contaminants in the water. There's like a whole lot of other factors, but I think temperature is a pretty obvious factor that is really linked to declines in the field, which is why we're starting with that. Yep.
Have you been able to see or like work with anybody else who might know, like you're working with the kelp and see if the kelp is declining, but what about all the other species that like need that? Like, what do you know about what the effects of that are?
Yeah, so the question was, if kelp are declining, what are the ecosystem level effects on all the other species? And yeah, that's a good question. There's a lot of groups doing active surveys and scuba surveys, underwater surveys, remotely operated vehicle surveys, and they are looking at like all the other invertebrates as well as fish. And I don't know if, I guess we don't have, I would say that if you lose the kelp forest, you likely lose a lot of biodiversity because there's a lot of species that live within kelp forests, but I think we're still trying to figure that out. The other important thing is that there's 26 species of kelp in Washington. So the bull kelp are the obvious ones that you see at the surface, but there's all these other short kelps called understory kelps that are living below the water at the bottom. And so those are actually not declining as much. And so you might still have a healthy understory kelp community that can still support a lot of species. So I think we still need to do more surveys and figure out what's happening. Yeah.
There's also a request online asking if you can tell us a little bit about your research with the kelp microbiome in urban.
Yeah, so somebody had a question about if I could tell a little bit more about my research with the kelp microbiome and their metabolism. So yeah, when I started my PhD, we didn't really know anything about, particularly with bull kelp, we didn't really know what species of bacteria were living on them or what their functional role in the ecosystem might be. So I did a bunch of DNA sequencing to figure out what species of bacteria are associated with kelp. One cool thing I found is that bull kelp and giant kelp each have their own unique microbiome. They have their own subset of bacteria that live with them. These bacteria are distinct from the bacteria that live in the surrounding seawater, but sometimes the kelp bacteria are present at low numbers in the seawater. We also found that the bacterial community colonizes the kelp as the blade grows out. So the blade grows a few centimeters per day and it's growing really quickly, and the bacteria settle onto the kelp, and the bacterial community gets denser and denser as the kelp blade grows. Another thing that we found is that kelp from the declining population at Squaxin Island in southern Puget Sound actually had very few bacteria. So we thought that they might have more bacteria, but they actually had almost no bacteria. So we think that bacteria are part of a healthy kelp forest, and we suspect that they're interacting with the kelp by using a bunch of the sugar that kelp release, because the bacteria, most of them need to take up sugar. They don't photosynthesize. So the bacteria are likely eating the kelp sugars. And then I also found evidence that the bacteria might be providing the kelp with a source of nitrogen in the form of ammonium. So the bacteria might be beneficial to the kelp. And I mean, it's sort of similar to like, we have a really important microbiome in our gut, and plant roots have a microbiome that helps fix nitrogen. And there's a lot of important relationships with bacteria that we're still beginning to understand.
One last, anyone else? My question is, how deep are the roots? You got 100, so how does it not wash away?
Yeah, it's incredible. I mean, the thing that anchors them is called a holdfast, and it's like just this really thick tangle of tissue, and it usually grabs onto like a rock or something and sort of anchors them. But sometimes it's not heavy enough, and you'll actually see them like floating, and they can like move around because sometimes the float brings them to other places. Yeah.
Okay, great, thank you. And next week, it's Wheel of Flower. So we'll talk about maps.
Awesome. Thanks for coming, everyone. Thank you.